Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen at any time. After the presentation, we will answer your questions from our question and answer window. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Matthew Lotti from Ultragenyx.
Thanks so much, Leah. And good morning / good afternoon / good evening, everyone, depending on where in the world you are. Thank you so much for joining my presentation. I'm a senior research associate at Ultragenyx Pharmaceuticals. Our company is primarily developed towards developing treatments for rare or ultra rare diseases that oftentimes don't get focused on by larger pharmaceutical companies. My particular lab is located in Woburn, Massachusetts, on the east coast of the US, and we specialized in the gene therapy application to treat those rare diseases. And so today I'm going to be walking you through the development of an AAV, which is an adeno-associated viral, which is a vector we use to essentially deliver our therapeutic genome into patients. We're going to be talking about an AAV automated capsid titer, essentially assessing the concentration levels of these AAVs, these adeno-associated viruses that contain what is essentially the treatment, using a Gyrolab xPand Immunoassay platform. We're starting to automate our samples as much as humanly possible because my role is within analytical development. So we're both doing the testing of samples that come down the manufacturing pipeline, as well as developing more and more assays to both quantify and nail down the biophysical aspects of products in development, but also run those samples using those same methods. The problem then becomes is the more methods you develop, the more samples you wind up with in your lab. So it becomes incredibly important to increase throughput over time. So this is a bit of a hybrid presentation where we're going to be talking about development, but there's going to be a heavy automation slant to sort of get samples tested in as quick a manner as possible. Jumping a little bit into an overview about what my company is and what we stand for, we're basically focused on looking at diseases that don't currently have treatments or even research underway. A lot of the patients under these treatments possess rare diseases, and without a dedicated mission to really focus in on tackling these, they would largely go ignored. And this is really summed up well in the words of our CEO, Emil Kakas. We're really looking to develop the first ever approved treatments for these rare diseases. And not only that, but we also want to share them as quickly as we can with the global community. So you'll find that our development timelines are essentially in a state of constant acceleration. And we try to have as much sharing and collaboration, not just with other scientific entities, but also a lot of our patients to really get them up to speed and get them into trial programs as soon as possible. Now, fear not, I am not about to read the wall of text on the slide you see before you. I consider that a pet peeve of mine when a slideshow has a gazillion words of text on it. This is being recorded and these slides will be available afterwards for you to review in detail. But what I'd like to really emphasize in these next two slides, I should say, are the three pillars on which Ultragenyx stands. The first pillar we've already talked about. It's that of the 95% of rare diseases that exist in the world, of the rare diseases that exist in the world, more than 95% currently have no treatments available, essentially leaving the individuals afflicted with these diseases with few options. Our first pillar is to not only focus in on these diseases, but also to accelerate the process of their delivery as quickly as humanly possible. A lot of the time, the average number of years it takes for a medication from entering clinical trials to approval can go anywhere from 7 to 8 years. When it comes to larger pharmaceutical agencies, we're gunning for a 5.5 to 6 year turnaround time, really trying to accelerate this process throughout. The second pillar of what we're doing is we're looking for patients to have access to these therapies as soon as humanly possible. I already talked a little bit about the speed component, but now the accessibility component as well. Not only making sure that these medications are priced fairly, but they're distributed to as many areas in the world as possible. And then finally, the collaborative pillar, the pillar of supporting the overall rare disease community. This is actually two-pronged. The first prong is in working with other scientific industries to share our knowledge and methods to support their developmental efforts to basically get more medications pushed through the pipeline as quickly as possible and address these issues. The second prong of that is actually working with patients, individuals who often feel like they don't have much option, don't have much choice. There's not a lot of other research being going on to center in on their rare disease. We actually work and advocate with them, try to help get them into clinical trial programs, but also give them support throughout the process and help them in whatever way possible. And it's these three pillars that really drive and motivate us. Now, as I mentioned early on, my particular wing of Ultragenyx works with gene therapy. And so I'm going to be talking primarily about the development of Capsid Titer, viral Capsid Titer. These are going to be the viral components that we have now removed the adeno-associated viral genomic DNA from, and instead inserted DNA that is of our therapeutic treatment. In the course of doing so, I'm going to be talking about the what. What is the gyro, the mechanism I used to develop this? What are the backgrounds and the benefits of using this system? The where, how about internally does this system work? What is its overview? The how, now we're jumping over into the actual assay development side of things. And then finally, the why. What other applications might this gyro and this method have in terms of expanding out to other assays? I had mentioned that the throughput is only going to increase in time. So the more flexibility we can get out of this device, the better. Starting off with the benefits of and the background of our automated system, I've already mentioned we're going to be using adeno-associated viral capsids as the primary vector through which we're going to be delivering our therapies. Now, when it comes to determining final capsid concentration, once you get to drug substance, we're going to be using methods like UV or MADLs to really come up with an accurate assessment there. The problem is that when you're working with samples that have very, very low capsid titer concentration or potential impurities, and you're going to run into both of those instances throughout the manufacturing process, really up until the point where you have enough clearance and purification to where you're getting that drug substance, is that these methods aren't really applicable at that point. You're either going to be getting concentrations that are too low to effectively monitor and get an accurate idea about the concentration of viral titer that's in there, or there's going to be impurities that give you false positives. So to get around this and to ensure monitoring and product consistency throughout manufacture at these earlier steps, we had initially used commercial antibody ELISA-based kits to determine AAV titer. The problem with these kits is that they were quite costly, they're time-consuming to run, and as we're going to talk about in a little more detail on my next slide, they required a very, very large pre-dilution within all samples tested therein in order to actually fit the standard dynamic range on these plates. Now, this table sort of breaks down the comparison between the ELISA kits we were using and the Gyrolab xPand system that we've kind of migrated over to. When it comes to the overall timing, running an assay is now about half the time that it was to just run a standard ELISA. The sample volume levels used are the same, but if you'll notice under the Progen ELISA kit column, that's due only to a higher dilution factor, and I'll touch base on that when we get to the bottom of this table. An overall sample capacity, you actually wind up with 112 channels versus 96 on the Gyrolab system. I can actually run more samples per run. And all of these factors essentially combine to make each sample tested on the Gyrolab xPand about $10 cheaper than that of the ELISA kit. And considering how many hundreds, and it's growing rapidly to thousands of samples we're going to be having in our pipeline, this can actually be an incredible cost savings. Now, finally, when it comes to assay dynamic range, this is what I mentioned before talking about that 4 microliter volume up above. You'll notice for the Progen ELISA kit, the dynamic range starts at 8E6 and goes down to 5E8. Oh, I'm sorry, I had that reversed. It starts at 5E8 VP per mil and goes down to 8E6 VP per mil. For a lot of our products that are about the drug substance area, you're going to be seeing concentrations at about 1E13 or even 1E14. Once you actually are in the manufacturing process, when you're going to be having samples that are not as highly concentrated, you're still often going to be having samples that are about 1E12 and even 1E13 at times. What this effectively means is that in order to take these samples and have them fit a standard curve that starts at 5E8, you would have to do a pre-dilution factor of sometimes up to 1 in 1 million to even have that fit onto the ELISA kit curve. At which time, when you have to compensate for that dilution factor, your accuracy could potentially suffer as a result. When it comes to the Gyrolab, however, we were able to effectively develop a dynamic range that starts at 5E12 and goes all the way down to just about 2E9. This requires much fewer levels of pre-dilution, if at any pre-dilution, helping to ensure accuracy throughout testing. Now, when it comes to the overall automation overview, the beast of a machine you're seeing on the left-hand side of the screen is our Gyrolab xPand. It is a 5-disc system, and as you can see, it takes up an entire bench. Myself and my colleagues have nicknamed it Godzilla for its gigantic size. The picture you see on your right is actually not fully accurate to this system on the left. This is actually the internal view of the one-plate system. And the reason I've included the one-plate system is that it essentially shows us everything we need in tight detail rather than having it all spread out. The real benefit of having a 5-plate system is it can run multiple assay discs at sequence in a given time. You essentially can get a greater level of throughput out of it. And what's really nice is it has the flexibility to have each of these discs not necessarily be the same assay. For our intents and purposes, we're running AAV8 Capsid Titers, but we could just as easily follow it up with an impurity disc right behind it, and the gyro will simply pick it up in sequence. What is important to know about this system is that its liquid handling is only functional when it comes to taking liquid out of the dilution plates and adding it onto the CD-based disc, which in this case essentially works like a 96-well ELISA plate. It's going to be the platform on which this reaction takes place, and this platform on which the reaction is then going to be analyzed and quantified off of. You still have to take the time to manually make your standard curve and dilute out your samples. So there is potential for an additional liquid handler to be added down the road for this initial step. But at the moment, we're just essentially using pipettes to put together this dilution plate. The benefit to this is it does make the learning curve a bit easier for those who are kind of well accustomed to putting together ELISAs, because really the only thing that's happening now is their dilution plates are being liquid handled by the gyro, added to the gyro discs rather than into an actual ELISA plate. And from there, the gyro effectively takes over. So if we look at the actual mechanism of the automation, we're going to be working our way from left to the gyro disc is actually made up of sections of microfluidic channels. Depending on the disc type, the volume of sample injected into each of these channels can vary. You'll see on the right, we can go into 20 nanoliter volumes, 200 nanoliter volumes, or 1000 nanoliter volumes. So we're actually using a very small amount of sample throughout, which is a very good thing, because the more sample we can save, the more we have on hand for future testing. Look closely at these channels. We're going to look at the image on the right now. You're going to see that right in the middle, there's that little slot where the gyro will actually insert the sample itself. It will then have areas that it actually adds the capture reagent and the detect reagent. What I really want to draw your attention to, however, is this capture column, the very bottom of the microfluidic channel. If we zoom in on that capture column, we're going to see these little streptavidin-coated particles in series. Now, the best way I like to describe this as essentially an upside-down sandwich ELISA. In a lot of sandwich ELISAs, you typically end with a streptavidin-coated, often detection reagent that is then going to have a colorimetric reaction with TMB. Here, we're using the streptavidin as almost an anchor. These little coated particles are going to be using this streptavidin to then interface with a biotin-labeled capture reagent in this case. Now, within the disc types, you can have high capacity discs, whereby you have a greater amount of streptavidin coated on individual particles, allowing you to effectively capture more of the biotin-labeled capture reagent than you otherwise would. But it varies depending on assay type, and we'll look a little bit more into that as we get into the development. You'll see we use a couple different disc types to get the best results. Once you have this conjugated biotin capture reagent attached to your streptavidin-coated particle, it is then necessarily going to be capturing the antigen out of the sample as any other ELISA would. Just like a sandwich ELISA, you're then going to have detection reagent that caps off that antigen. And this detection reagent is also going to be conjugated onto, in this case, what we'll be using to detect the signal. In this case, we're actually going to have a conjugation onto an Alexafluor labeled dye. And the fluorescence given off by this dye is going to correlate to the amount of concentration, I should say, or titer of viral particles, and then be interpolated off a standard curve for analysis. So these initial experimentation, this was actually done by Ellen Lee from Gyro, big shout out to Ellen, and another colleague of mine at Ultragenyx, Shinji Oyama. What they worked with here was this initially came within a self-contained kit. This AAV8 Titer kit came with a 1,000 nanoliter disc and a single dilution of the detection reagent conjugated onto that Alexafluor dye. Now, initial testing wound up maxing out the signal when this dye was not diluted out. So some of their testing actually broke it into 3 different dilution types, a 1:3, a 1:9, and a 1:27. And those are the 3 curves you're seeing on the graph on the right-hand side. They also shot for a standard curve range that they would figure would fall more or less in range to what we'd expect. Once again, we're going to be expecting 1E13 to 14 for drug substances, and then 1E11, 1E12 for more manufacturing-based samples. So they aim to have a standard curve that really captured these. And what the results were found were actually very, very encouraging. What you're really seeing here is we have a solid R-squared for all 3 results. At the varying concentrations, you're just getting higher levels of signal to background. So you're getting similar performance throughout. Essentially, any of these are usable. I would probably steer away from the lower dilutions just because you have a little bit more background there. But at the end of the day, the real question was, can we extend this range further up? Can we essentially get into the 13s and the 14s in terms of magnitude to prevent having to make these dilutions at all? So a very successful starting experiment. Now it's a question of, can we go higher? So our second experiment to compare against the 1,000 nanoliter disc brought in a 20 high capacity disc. Now this disc is only being loaded with 20 nanoliters of sample, but it has that high capacity characteristic that allows the streptavidin beads to capture more of the capture reagent than they otherwise would. After running this with a higher standard curve that started at 5E13, what we found is we had a good amount of repeatable precision amongst our replicates, used on these standard curves, but our bottom end of the dynamic range completely dropped out after 1E12. This, unfortunately, was not going to be usable because we have samples that would be coming in around that level. So we got the top end, we lost the bottom end. The next step then became to try to get this lower end of our dynamic range back. So we devised a multi-parameter experiment, basically the shotgun approach. We're going to put together a web of multiple disc type, buffer types, and concentrations among that detection reagent to spiderweb together which little crossroads work together best and get us a sweet spot. In terms of the 2 discs we used a 200 nanoliter disc. We had not tried using this disc type before, so this was new territory. And we wanted to compare it to the 1,000 nanoliter disc, which we did see that good upper end on initially. The 20 high capacity disc was not brought into testing here simply because not having that lower end available essentially removed it. But I will show a graph of it on our next slide for comparison's sake against the 200 and the 1,000. Insofar as the buffer types, we were fortunate in that the Gyrolab AAV8 Titer kit came with its own buffer type, but Gyro does offer other buffer types as well for different situations. So we tried bringing a Rexit buffer, one of those alternative types, into play as well. And then finally, we broke up our concentrations. For the 200 disc, we hadn't done any other experiment looking at the different concentration factors. So we started with a neat concentration factor versus essentially a low dilution of only 1 to 3. And then when they came to the 1,000 nanoliter disc, again, we started with a neat concentration, expecting it to not do well, but wanting to put it on there for comparison's sake. And then holding up against a 1 to 10 dilution. As you saw prior, that 1 to 9 dilution kind of was right in the middle there, where its noise level wasn't too high. So this is what we wound up going with. All right, now, I understand there is a lot of data on this slide. So what I'm going to do is we're going to walk our way from left to right, and we're going to do so by me pointing out certain bits of data. On each of these graphs here on the left, you're going to be seeing 4 standard curves, each one representing a dilution of the detection reagent, and B, a buffer type. So cross those two variables together, you wind up with 4 curves. When it comes down to the results of the 1000 nanoliter disk, although all the curves had a good R-square valued, as you can see by the replicates being all over the place and scattered within that circle, we had overall low precision and accuracy among all curves tested. Essentially, in this case, we're losing all ability to accurately and precisely get results once you drop past 1E10. So this essentially was nixed from consideration. Once we moved over to the 200 disk, however, things got a lot more promising. Now we're seeing a lot more tightening up among our replicates at the bottom ends of the curve. And you can see at the top of the curve, we're still where we want to be. So it's really just about nailing down these final 3 points. Once again, all of our curves came in with a solid R square. But when I really zoomed in and took a look at these data, what I really found was we only had good precision and good accuracy among 1 of the 4 conditions tested. Fortunately, that left us with 1 optimal condition. So what you're seeing here is essentially the percent CVs or overall precision among each of the combinations tested. You'll note that column in green, which is the kit-based buffer at a neat detection level. So what the kit that came with the buffer was used and no pre-dilution was made on the detection agent. This actually produced the best overall CVs and precision throughout the entire process. And you can see as you work through the other parameter combinations, you are getting levels that rise up to pretty high. And this also extended into our accuracy as well. You do have a little bias come into play at the utmost level of our standard curve, but that's something we can work with. You know, if we really need to go down to, let's say, 5E12, that's only going to require a very, very small dilution factor. So losing that isn't too big of a deal. What is, however, very promising is you can see how much the accuracy improved towards the bottom of the curve, going all the way down to 5E8. This essentially established the condition that we wanted. And then finally, over here, all the way on the right, we're going to be looking at our 20 high capacity disk. This is the data we looked at a short time ago, just so you can compare the curves against each other and see that with this, you're getting overall precision and accuracy tanking once you get past 1E12. So here we are. We have a good solid, what we think is the most optimal condition, the 200 nanoliter disk, the sample buffer that comes with the kit, and the use of a neat detection concentration. The next step then became actually taking our samples, which you'll see at the below, as sample type A through sample type E. These are essentially representing the manufacturing process from start to finish. Imagine sample type A being on the level of clarified harvest or things that are just coming out of the bioreactor, all the way down to E, where now you're getting close to a final product. The goal here was to actually run these samples on this against this particular standard curve, noting what precision they have, noting to see if they have dilutional linearity as you make multiple dilutions, and also getting an idea what your limit of quantitation were. And the big thing here was actually going to be how do you interpolate these sample results off of your standard curve? What results do you get? Now, if you'll note our standard curve graph over here on the right, you can see towards the bottom, there's a little bit of a separation among our replicates. That was a bit of a concern at first. However, the Gyrolab has multiple maintenance methods in between runs you can use to clean this up. Specifically a method called needle desorb, which of the small amount of liquid handling done on the gyro, it works with very, very small volumes. We're talking nanoliters. So even little bits of cleaning can go a long way to ensuring accuracy. In terms of the sample results were very, very promising. Within each sample replicate, these were all run in triplicate for each dilution factor. So essentially that first CV column you're seeing is going to be just the percent CV for that dilution at that factor within a sample. So essentially we have intra-sample CVs precision and inter-sample CVs precision once you combine all those dilution factors together. And in both cases, we came out with really strong results where all CVs were under 10%, or actually under 9% for all cases tested, which was excellent. This is a very, very encouraging result. We also noted dilutional linearity across the samples as well. So this was very, very encouraging. And we were able to move forward with this and now begin testing all of our Capsid Titer samples regularly. But because flexibility is such an important component here, we also wanted to ask ourselves the question, can this be used potentially for other applications? So we got to thinking, our Capsid Titer method measures all capsids within a given sample type, be they full capsids, be they capsids that are only partially full, or even empty viral capsids. When we run our qPCR method, we're going to be getting the titer of only the full viral capsids. We asked ourselves the question, if we took a ratio of those full capsid viral titers and divided it by the complete capsid viral titer the Gyrolab gives us, could this essentially act as a surrogate measurement for an empty full capsid population? Now, this is where we kind of turn around and look at analytical ultracentrifugation, which we typically use on a lot of our drug substance samples to finally quantify the level or the percentage of what capsids are full, what capsids are empty, what capsids are partially full. We asked ourselves the question as, could this ratio serve as another way to go about doing this, or to at least produce this sort of results? Would there be correlation between this ratio and what we are getting off of our C results? And if we look over here on the graphs at the right, the one I really want you to point your eyes to is the one closer to the very bottom, where we're actually taking this PCR to gyro capsid ratio and comparing it to AUC ratios of the full cells. And what we're actually seeing here is starting to get a little bit more correlation as we go. So this is a very, very early on step in the process. And as I'm going to talk about more in an instant, it's not something we're going to be able to use very often, but it does have a potential niche application that could be useful if we were to continue moving forward with it. Now, when it comes to the use of this particular comparative study here, one thing you need to bear in mind is that we have three assays being kind of compared against each other. We have our qPCR for overall full capsid titer. We have the gyro AAV capsid titer, which gives us everything. You're going to be getting full, empty, and partially full. And then finally, we have the AUC, which is going to be giving us a ratio breakdown of this percent is full, this percent is empty, this percent is intermediately full. Each of those assays is going to have an inherent level of variability baked in. All assays do. And because you're now putting all of those assays together into a single formula, you're going to have a compounding of that variability. That factors it into play. As such, we are still going to be using for our drug substance and products that are further down the line, methods like AUC in conjunction with UV and MADLs, rather than this ratio that we talked about here of your qPCR titer divided out by your gyro titer. However, there's another sample type where that particular ratio can be handy. And this goes all the way back to the beginning of the presentation, whereby you want to make sure you have consistent titer throughout the manufacturing process, even in samples early on that might have very, very low concentrations or very, very low volumes, and that are still high in impurities because they haven't gone through the filtration process yet. This is a potential application towards those samples, giving you an idea of how much is empty versus full before they're even at that step for the AUC. And this can be used to make modifications potentially in various column studies when it comes to manufacturing. If they find one column isn't working very well, they can then stop and try to reassess before they make their way that much further down the pipeline. This is still very, very early on, and I know its applications are limited. But if you look over here at the graph on the right, one study done by a colleague of mine, Wunyun, she found that when it comes to actually looking at the gyro capsid titer on this particular column, you're having breakthrough long before you would if you were just taking the overall qPCR titer, what is essentially the titer of the full capsids, into account. When you take the overall titer of all capsids into account, you're seeing this column gives way a lot earlier. And depending on what part of the manufacturing process you're in, the overall titer could actually be a lot more accurate, depending on how much of it was still left, depending on how much was still filtered out. So it's very, very useful for the manufacturing process as well. And we're still going to be diving into how much more application we can do. But for the moment, we're thinking that this can not only provide through column breakthrough studies, but also potentially clone and media screening for higher full capsid titers. So in conclusion, when it comes to the Gyrolab xPand compared to the ELISA kit, we're seeing overall higher accuracy and precision, a larger dynamic range for our assay. It actually comes out to be lower cost per sample at a faster time and a higher capacity. The machine has the flexibility to run other assay types on it. So as we work our way into 2022, we're going to be developing a lot more manual assays directly onto the gyro, which, because it's a 5-plate system, can then be run in sequence. We have some potential correlation to biophysical measurements, allowing us to quantify essentially whether or not capsids are full, intermediately full, or empty via this ratio with early samples in the manufacturing process, even though this is very, very new. That said, I want to give a huge amount of thanks to not only my colleagues at Ultragenyx, but also those at Gyro, especially Ellen Lee. She was so instrumental in helping us learn how to use the gyro and develop these processes. Not listed here, but who I also want to give a tremendous shout out to here is Cecilia Anarin from Gyro as well, who invited me to speak at this presentation, as well as Leah Rosin and Alex Nicolatis for helping set up this entire presentation as well. Thank you guys so much for your support. And that said, I'm happy to answer any questions. If you have any questions for me outside of this, please don't hesitate to email me. My company is growing like crazy right now. So if there's anything you're interested to learn more about, please let me know and feel free to reach out to me at any time. And I'm happy to take your questions. Thank you.
So for Matt, what other assays do you currently perform or plan to develop using Gyrolab?
Sure, not a problem. So currently, I developed this AAV Capsid Titer in parallel to an impurity assay. Essentially, an impurity assay of ours, which I developed in ELISA for, which took anywhere from 6 to 7 hours, a lot of time. I was able to reduce to only 2 hours. And with the same amount of accuracy and precision, and actually a greater dynamic range for a certain impurity within our products. And a lot of that development process mirrored what you saw there on the, Capsid Titer process. The only difference was we got very lucky with that AAV8 Capsid Titer process because Gyro offers AAV8 Titer kits that essentially come with baked-in materials you can use to put that together. It falls to you to provide your own standard curve and then find, the correct disk type and the correct dilution level necessary for the detect and capture reagents involved. But when it comes to you building a kit from scratch, which is what we essentially had to do for the impurity ELISA, we had to provide our own primary and secondary antibodies, conjugate them ourselves to the biotin on the capture side and the Alexaflor on the detection side, in addition to providing our own standard curve. But ultimately, we were able to extend the upper end of the dynamic range of that assay. So worked out very, very well. And we're going to be actually taking at least 2 or 3 other impurity assays of ours in early 2022 and putting them on there.
Okay, so this is probably the last question we have for this webcast, but like Matt said, you can go ahead and send follow-up questions to him, and he'll get back to you. So do you plan any other optimizations for this assay?
Definitely. I mean, the funny thing about this assay is we've been running it steadily, but it is still very, very early on. One of the first things that comes to mind is I'd like to develop a positive control for this assay just to give us a better idea of its accuracy, especially over time. Monitoring a positive control over time, monitoring a positive control's stability over time can give you an idea about whether or not any changes you see in that positive control's performance could potentially also be global and affecting everything else. I mean, you run into the trap of, is it the control itself that is the ability over time, or is it more plate-wide? But I'm getting down a rabbit hole with that one. Essentially, I'd love to introduce a positive control over time. And if I am further able to extend the dynamic range in either direction, I would like to continue to, just to make life a little bit easier. To prevent matrix effects, I would always like to keep a 1 to 2 dilution at least in play. So I'd prefer not to put something on there neat. But if we could get things lower, it would certainly help things out at the time. Where we're at right now with their standard curve is where I'm seeing the best accuracy and precision. And when I try to go above and below, it looks like you're getting a little bit wonky there, but I'll keep tweaking it and see if I can get it a little bit lower or higher.
Okay, well, thank you, Matt, and thanks to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up link providing you with more details. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Thank you all.